Hi, welcome to the NTE Lab. I am the lab director. My name is Tracy Cui, professor of bioengineering at the University of Pittsburgh. In my lab, we are primarily interested in a variety of neural interface devices that are implanted into the nervous system for recording neural signals, stimulating neural activities, measuring neurochemicals, or sampling from the brain tissue or deliver drug into the brain tissue. For all of these devices, we need to implant the device into the neural tissue, but implantation induces a damage that can be observed from the two-photon microscope. This mechanical trauma causes prolonged pathological calcium response from the neurons, as well as activated microglia that send processes towards the device in the attempt to wall off the device. So our lab is interested in finding the cause of those host tissue responses and develop biomaterial strategies to reduce the host tissue response and in order to obtain long-term, high-quality, stable recording, sensing, or neurostimulation. So as you can see from our lab logo, we are at the interface between neural tissue and electrode. And the idea is to develop a technology such that the electrode and tissue seamlessly integrate and live in harmony. However, current designs of the electrode devices have a number of mismatches from the neural tissue. So the first mismatch is you have to transfer electrical signals across the electrode tissue interface, which has a high interfacial impedance to which can prevent getting high quality signals or adding a lot of barriers for charge injection. To deal with this, we have been developing conductive polymers, which is really a platform technology where it allows you to tailor the conducting polymers directly at the electrode surface. And by incorporating nanomaterials, we can offer a variety of functionalities to the electrode. Primarily, we can obtain high quality recording because of the low impedance as well as high quality stimulation. So currently we have an NIH Brain Initiative project looking at different types of advanced conducting polymer and nanomaterials in order to understand the stimulation efficiency and safety from those materials. The second mismatch is the mechanical modulus mismatch, where inserting very hard device into a very soft neural tissue that causes a mechanical injury, and also this constant irritation will lead to inflammatory tissue response. So to deal with this, we have been developing brand new materials that are soft, elastomeric, and conducting. So using these materials, we have made nerve electrodes, muscle electrodes, and brain electrodes, and we've shown that they are very compatible with the neural tissue. So currently we're developing a peripheral nerve electrode under a NIH STTR grant in collaboration with TDA Research. The last mismatch is at the between the bioactive neural tissue and the non-bioactive electrodes. And the bioactive neural tissue recognize electrode as a foreign body and try to generate a lot of toxic chemicals and also cause inflammatory response that would wall off the device and damage the neurons. So in order to deal with that, we've been working on strategies. The first strategy is to develop biomimetic coatings to modify the surface with anti-fouling molecules, antioxidant, as well as neuroadhesion molecules and anti-inflammatory peptides and proteins in order to modulate the cell response to the implant. So some of our coatings has shown very good result in terms of improving the recording quality and stability. Currently, we are working on a Brain Initiative U01 grant, which is aimed at improving the stability of the bioactive coating in order to make this coating technology available to the general users in the field of neuroscience. The second strategy is to actively deliver bioactive molecules to minimize the inflammation or promote neuronal health. To do this, we are using a electrically controlled drug delivery technology. In this case, we can load the drug on individual electrodes and trigger the drug to release by applying the electrical potential. So this, as you can see, allow us to precisely control when, where, and how much drug we want to release. So in addition to using this technology to improve the tissue electrode interface, we can also use this technology to modulate neural activities. So here in this case, we are working with Carnegie Mellon University to develop a multimodal neural probes that can not only record, but also image the cell types, as well as applying neurochemicals in order to identify the types of neurons that we're recording from using this electrically controlled drug delivery technology. Finally, we are working on neurochemical sensing. Some of the technologies are that utilize electrochemical sensing. So with our PDOT-based nanocomposite, we've been able to hugely improve the sensitivity of neurotransmitter sensing and enable both fast phasic release detection as well as slower basal tonic level concentration measurement within the brain. So the next step is to move those technologies onto multi-electrode arrays. So here's an example of our flexible multi-electrode array device on which we will be modifying the electrode sites so that we can do multiple measurement from different regions of the brain, as well as measuring multiple neurochemicals. This work is supported by the National Institute of Drug Abuse. So as you can see, our primary research goal is to understand the biological response and how they affect neurointerface function. We use multimodal approach, including electrophysiology, 2P imaging, molecular biochemical tools, as well as materials and explained characterization to understand the failure mechanism of neural devices. And then with the knowledge learned from this fundamental study, we will guide our material science and device design. And using those platform technologies, we can modulate that response. So NTE Lab is a big, happy family. We always welcome talented and motivated students to join the lab. For more details, please check out our website.